Hello, Feds for Freedom fans, supporters, members, or anybody just happening to scroll through their social media feed and see something they find interesting. I'm the Vice President of Feds for Freedom, Jim Erdman. I'm a former federal government employee. I worked at the CIA. And today we're going to be talking about the origin of SARS-CoV-2 with Dr. Stephen Quay. He's an expert in his field, and this is a two-part interview. We wanted to get the first part out as quick as possible, so we're putting it on social media. But you'll be able to find it on our podcast fairly soon. The second part will be coming out as soon as we can. But without further ado, I'd like to introduce you to Dr. Stephen Quay and let you take a look for yourself. Enjoy.
Welcome to the Feds, insiders bringing accountability, integrity, and reform to a broken bureaucracy. At Feds for Freedom, we value constructive dissent and healthy debate. The views and opinions shared in today's episode are those of the speaker alone and do not express the views or opinions of the U.S. government or any other employer.
I have about 9,500 federal government employees who are really interested about the science, where this all started. And for me, it did start with the science. I was, let's say, obsessed with SARS-CoV-2 when it hit the scene. It hit my educational background. It's the science that I've always loved. And so you showed up on the scene and I started seeing some of your tweets. I started seeing some of your social media. I think on LinkedIn was where I first saw some stuff. But I'd love to hear your background, who you are, and then maybe we can jump into some science questions.
Sure, Jim. So, I grew up in Michigan and over near Detroit for most of my childhood. And then in high school, my dad went from being a high school physics teacher to a principal over in Western Michigan. And then I ended up graduating from high school near Grand Rapids, going to Western Michigan undergrad, Michigan for an MD, PhD, and a master's degree. Then off to MIT, working with a Nobel Prize winner at night and doing my residency during the day at the Mass General. Then I taught at Stanford for a decade, invented the gadolinium that's used by MRI. It's been used about 60 million times. And then I've been doing a series of biotech companies and currently run a public company, ATOS. We're going to try to prevent breast cancer, which is pretty exciting. And it is my day job, and it's what really motivates me.
Well, then I definitely need to introduce you to one of the board members on Feds for Freedom, Dr. Ruddy, Kathleen Ruddy. She's been involved in medical work for cancer for a long time, and so she's on our board of directors. So afterwards, we'll figure out how I can get you in contact with her. And one other side note, I lived in Livonia, Michigan. I moved to Livonia from Cleveland, so we weren't too far away.
Yeah, no, I grew up in Northville. I'm actually writing a book. I'm going to write a book that's going to come out next summer. We're going to have a big event in Northville. So if you're in the area, I'd love to have you come back and do it. But basically, it's a book of Northville from about 1950 to 1980. And it's a book that presents it as if you're riding a bicycle, because that's what we did in those days.
Yeah.
So we have pictures of every house in the town. We have commentary on over half the houses. It's a really exciting project. That will memorialize it, because it was built by Henry Ford as one of his factory towns for one of his valve plants. So long story there, but anyway.
My dad was a Ford guy for 30 years. So
there you go.
My dad put me through school. So
Excellent.
Well, to jump into this, I think for me, what was fascinating early in 2020 was the discussion on whether or not this was natural, zoonosis, or lab leak. And can you talk in general terms about sort of the gaps or what makes it hard to come to a conclusion? Like, it's really hard to come to definitive conclusions, but can you talk about what makes it hard to come to a conclusion like that?
It depends on whether you're at 30, 40, or 50,000 feet to describe what makes it hard. Probably, at 80,000 feet, if people are not sharing data that they have for whatever non-scientific reasons, and that parentheses political or whatever else you want to put into that bucket, you're going to have trouble. But I've approached it. I'm finishing a book on this, but I've approached it as a forensic exercise. Basically you have somebody who's disappeared. You don't have a body. you have, you know, a guilty looking husband, you got a lot of circumstantial evidence, and then how do you build a case in that particular setting? And so that is the approach I've taken, which means that I'm mostly using information inside the genome itself to tease out. It's basically, you know, the virus as witness is kind of the concept there. But okay, so backing up, if you when you have a new outbreak of a zoonosis, so what's a zoonosis? It has three elements. It has the word zoo in it, so it means there's an animal involved somewhere. So a zoonosis is the three factors: is an animal infected with a microbe that then gives it to a human. The whole Twitter version of the crux of is it a spillover from nature or did it happen in a laboratory is where was the animal when the human got it. So if the animal's in a market, it's a natural spillover. If the animal's in a laboratory, it's a lab leak is what they got calling it, which is a pejorative, frankly. I don't use it, I mean, because it implies, you know, plumbing problem or something. It's like, you know, your toilet overflowing, but it's not that at all. It's a person working in a laboratory who doesn't even have to be careless, because SARS-1 infected somebody in a BLSA-4, which is the folks with the spacesuits and the air tanks. So you're BLSA-4, you're working with SARS-1, and you're the last person to get SARS-1. So in any case, the answer is where was the animal when the person got infected? And so look at it, when it happened in January 2020, you know, we first heard about it in January 2020. We all thought it was from a market. It makes sense. And the reason it makes sense is that there are, when you do an analysis of how many natural spillovers there are in Asia every year and how many lab leaks, it's 8-to-1. So there are 8 spillovers of something. Sometimes it's a virus we've already seen, sometimes it's a new one, but there are 8 a year in Asia versus 1 lab leak in Asia. And that's again from peer-reviewed literature. So you start with an 80, you know, what's 8, you know, 8 divided by 9, you know, it's about 88%. So you start with an 88% probability it came from nature. You know, only 12% from the laboratory. And then, but then you have to build from there. And, you know, this Bayesian analysis I did, it's up on Zenoda and it's been viewed, I think, 250,000 times. So I got 390 publications and it's my number one of all the stuff I've done in my whole life. But anyway, we start with that 87% or 88% likelihood that it came from nature. I actually, because of other factors, I bumped it up to 98 from nature. But then by the time you go through 26 levels of evidence and run what's called the Bayesian theorem, which is a mathematical way of taking prior information and inputting it into a subsequent decision tree of is it A or is it B, you end up with 98% probability from a laboratory.
Thanks. I was just about to ask you to explain Bayesian, but you took that. And the other thing I'm really thankful for is you gave me a nice broad description that sort of gives an idea, and it makes me feel good because the questions I've prepared, I think, follow along with some of the things you've mentioned.
Okay. Yeah.
So, and I'll start from the very beginning. This is less science, but more your gut instinct. You know, and I think you sort of answered it partially. There was an influx in early 2020, and a lot of the papers that came out were really heavily focused on natural origin, zoonosis, right?
Yes.
And so it sounds like, from what you've said to me now, that's a natural conclusion if you've got an 88 or 98, depending on what the analysis looks like from the beginning. So in general, do you feel as though that was a normal occurrence for there to be a heavy preponderance of focusing on zoonosis early on?
No.
Okay.
And the answer is I'm going to be, you know, very particular about the words and that sort of thing. So what I described is that there are 8 zoonoses per year in Asia, right?
Okay.
So that's fact number 1. So that you start there and you get to 8812. Okay. Next fact. This outbreak was in a town called Wuhan, which most people probably hadn't heard of, but it's kind of I describe it as sort of the Chicago of China, and what I imply by that was at one point in time, Chicago was this hub of railroads and things, and raw materials came into Chicago and they distributed out to the East Coast, West Coast, and that's what Wuhan is. It's a distribution center for most of China, and so it's a very modern urban city. It makes, you know, New York and London look like they're out of Charles Dickens. Very modern city, 11 million people, and it's nowhere near the places where bats have viruses that have the receptor, the ACE2 receptor that binds to humans. So immediately you have this anomaly where, okay, it happened in Wuhan, and not in the rural areas. Where did SARS-1 begin? Well, it began down in the wet markets of southern China, where they culturally, have a much more their cuisine involves much more of catching and trapping wild animals. So that's where SARS-1 came from, because it was a spillover from a market. So that immediately puts you sort of in alarm. Well, okay, why a big city? There are 100 million, I misspoke. There are 100 cities, over 10 million in China. Wuhan is one of them. So why there? And fact number 3 then is the real killer for me that starts flipping things for you is, if you look at SARS research from, let's say, 2003, when the first spillover occurred, so 2003, 2019, you go to the Library of Medicine and you ask, how many papers were there on SARS viruses in humans in that time frame, 03 to 19? And how many came from the Wuhan Institute of Virology, which is a study center in Wuhan, and it's 65%. So we now have a fact that 2/3 of all the SARS coronavirus research in the world happens in Wuhan, and that's where the spillover occurs. So now you're starting to get, sketchy on that. So those 3 facts, you go from, okay, Asia, it's got to be a spillover probably. Wuhan, well, that's a little strange. Let's downgrade that a little bit. And Wuhan is the Vatican of, coronavirus research, if I had to put it that way.
Yeah. Well, it's funny, this leads right into my next question was, so you said 65% of the research was occurring there. That's a pretty big number. And so the next question I had was there evidence of sort of researchers doing like genetic work on coronavirus viruses at Wuhan Institute of Virology prior to the outbreak? So do we have like there? I remember reading some stuff from, I think it was Dalglish and Sorensen, and they had laid out some of the research that was going on at WIV between, I want to say it was 2008 and 2019 or something like that. And they had laid out a pretty convincing argument that there was indications that the research was focused on editing, let's just say, of coronavirus. You're saying there's 65%. What can we draw from the type of research that was occurring there?
I mean, Wuhan Institute of Virology, which was started in 1950 with the Pasteur Institute in Paris as its model, was basically a center that was doing what I would call Darwin-like research, where Darwin took a boat around the world and collected samples and came up with the origin of species, his theory on evolution. And so from the mid-50s until 2003, the Wuhan Institute of Virology was primarily looking at natural viruses, collecting a lot of samples from the wild, bringing back to the laboratory, cataloging them, studying them, much like a natural history museum of virology, if you could imagine that. And then SARS-1 hit, and they pivoted very greatly to say, hey, we need to focus on not just all the viruses that can happen in China, but specifically human pathogens, because that was a big wake-up call. Coronaviruses had been around. There are 5 coronaviruses been around since 1935. They're one of the common colds, and they cause pretty mild disease in humans. But SARS-1 was about a 10% lethal virus, which I'll get back to in what that meant in January 2020, because that number 10% had implications on public health policies. But anyway, so starting then in 2003, and then also more in 2008, there were, in the parallel universe of genetic manipulation of viruses, there's a laboratory in North Carolina run by Ralph Barrick, who trained at USC with a gentleman named Dr. Michael Lai, it's L-I, he's sort of the grandfather of coronaviruses. So the virus has 30,000 letters, it has 15 genes in a particular order, and Michael did that. So Michael's at USC. He's studying coronaviruses. Ralph is his postdoc. Ralph's wife is the secretary for both of them and that sort of thing. SARS-1 hits, and Ralph is Taiwanese. So he gets brought back to Taiwan to help marshal the public health policy around SARS-1 as a coronavirus. Ralph goes back to North Carolina, where he grew up and was a state swimming champion, to the University of North Carolina, and continues the research and becomes, so if Michael's the grandfather, then Ralph is the father of genetic manipulations of coronaviruses. They're one of the largest viruses, 30,000 bases. Influenza is only 14,000. So and about 2008 through 2012, Ralph's work was the first to make a synthetic coronavirus that could replicate. What that means is that he took a natural, typically in the laboratory, what you do is you take a natural virus, you throw it on cells, it invades the cells, kills them, and then, a day or two later, you have a soup that has, a billion virus particles. You put one in, you get a billion in 2 or 3 or 4 days. And what Ralph did was he took the 30,000 letters, he broke them up into 8 pieces of about 6,000, I misspoke, about 5,000 each, and then grew each piece in a bacteria. And then threw them in a test tube, and they spontaneously assembled. And then he took that artificial piece of now 30,000 letters of just raw RNA, and he did what was described in the book Frankenstein. He used electricity to drive it into cells, and this naked piece of chemistry, this naked piece of RNA, went into a cell. Electrically, the electric charge drove it in. 2 days later, virus particles came out. So he created the virus from chemicals on the shelf. And that was the largest virus that had ever been done. So
Now,
continue.
Oh, sorry. No, please continue. I'll ask.
So continue into 2015, you begin to see collaborations with Dr. Shi from the Wuhan Institute of Virology and Ralph in approaching, in using the synthetic tools of biotechnology in the coronavirus space. So they took a MERS virus that wouldn't infect very well. They did serial passage, and 4 mutations caused it to infect cells very intensely. Now that's pretty dangerous because it's 30% lethal. MERS is 30% lethal. And in their paper, I think I put this on Twitter at one point in time. They're geniuses, but they're not wise, in the sense that they do this experiment to take a MERS virus and make it more lethal by putting 4 mutations into it. They know what 2 of the mutations do, and in their paper they say, we don't have a clue what those other two mutations do. So they're playing with a 30% lethal virus, and they don't even understand 50% of what it does to make it jump from species to species. And then, fast forward to 2017, 2018, they start to focus on the spike protein and using that as a Lego from, remember, Wuhan Institute of Virology has 20,000 viruses on the shelf because they have the Natural History Museum of viruses. So 20,000 backbones, and then they're just taking the spike protein and switching them in and out. And they describe doing that synthetically in papers and in grant applications. And then they describe putting furin cleavage sites in to see what those do. So when you end up with a virus in spring of 2020, that looks like the end product of 10 years of deliberate research in the laboratory, including serial passage, including genetic manipulation, including switching parts in and out, including making them more infective by putting furin cleavage sites in, which have never been seen in this class of viruses since William the Conqueror came across from Normandy into England in 1086. So you start to change your probabilities on whether this came from the Wuhan Institute of Virology.
Thank you. You hit on a number of things that I've got listed here. This is great. There are a few questions based on what you just said.
Yes.
And I'll try and curate the rest of the questions since you've touched on quite a few. Can you talk about how hard it is? Like you talked about Ralph Baric creating the Frankenstein, the RNA. Dr. Baric, clearly very talented, right?
Yes.
As I understand it, RNA is a very fragile substance. And to get that in, can you talk a little bit about just how hard it is to do that process? And now we're talking about RNA that's been stabilized for use in vaccines and things like that. But at the time, that was a difficult task, correct?
Yes, yeah, Jim. It's like so many things in science. The first person to do something, the first person to blaze the trail and chop the tree down and make a road, basically, does all the heavy lifting. And there's a lot of inventive work there, and a lot of what I call good hands work in the laboratory. But then at some point in time, it can become routine. So with your I guess CIA background, you'll love this story. So in either March or April of 2020, group of Swiss scientists, about 30 of them, put this paper out there. They bragged about it. So what they did was they had a way of growing the pieces not in bacteria and assembling them, but in baker's yeast. So the sourdough bread
Yeah.
stuff. So they said, okay, look, for $5,000, we sent off 8 pieces of this of SARS-CoV-2. We got them back in the proper format to grow them in yeast, and within a week, and we probably could have shortened it if we'd done a few things better. And within a week, we were able to get SARS-CoV-2 to grow in baker's yeast, and it's a higher yield process. It's better than bacteria because the yeast will put the sugars. Will glycosylate the protein, the virus capsid, more like a human will. So it makes it more effective as a product. And they were bragging about how they could have done it faster. But it's 5,000 folks, 30, I misspoke, 30 folks in tennis shoes, $5,000 a week's time. And they published this paper. The paper's been downloaded 110,000 times.
Holy cow. Really?
So I don't think there are 110,000 coronavirus researchers in the world. And I've talked to my friends. I've worked a little with the State Department and with people in Congress. I said, why don't you guys get the list of the people that downloaded and just, check it for me, just so I can sleep better at night.
Mm-hmm. Well, that's part of my second question related to the difficulty of doing that. Are there proprietary, now that he's done it first, are there proprietary procedures that are just associated with, say, like the lab or with a company? Or so do those procedures, those methods for doing that become proprietary?
Well, so University of North Carolina has, I want to say 3 to 5 patents, and NIH has patents on this. So a lot of the steps that are currently used to synthetically work with coronaviruses have been patented. That becomes important if you're commercializing it, but usually research use exemption allows anybody in the world to use what is patented information. There are also what I would call, I guess a patent lawyer in my spare time or something, so I use all their terms because I have 90 issued U.S. patents and won a bunch of litigations. So
Wow.
The other aspect is what I think you're getting at is trade secrets. So there are things that don't end up in papers, they don't end up in patents, but they're kind of the secret sauce, or of McDonald's secret sauce, which can allow a particular laboratory to have advantages over other laboratories. But in some ways, at this point in time, probably within 10 years, it starts to move into the area where my daughter's high school could do this in the lab.
Gotcha. Well, that's a nice segue, because my next question had to do with the lab. And I think people know the answer to this. This has been widely published, but just for folks who maybe aren't following it, were the labs in China, were they at elevated risk for poor lab procedures, for safety issues, those sort of things?
I want to answer that in sort of 2 ways. So a little background foundation and then the particulars. So, it's been long recognized that there are different risks of self-infection or of spillovers from a laboratory based on what you're working with, right? So long ago, we've codified BSL standards, which is biological safety laboratory standards, 1, 2, 3, and 4. So 4 is the spacesuits with the air into the ceiling, and gloves, and double gloving, and showers in and out, and double doors with negative pressure, so that the air goes in if there's a leak and that sort of thing. That's BLSA 4. That's Ebola, that's Nipah, and that's in those. Level 2 is a dental office. So you're kind of wearing gloves, you might be wearing a mask, you might have a hood with a negative, laminar flow, but maybe not. 1 is almost, nothing, and then 3 is an intermediate. So, and we've established standards of where these viruses should be worked on. So again, Ebola is 4. You don't do anything with Ebola at 3, 2, or 1, et cetera. So the coronaviruses had, look at SARS was 10% lethal, and it killed about 1,000 people, and it infected, I don't know, 8 or 9,000. But nonetheless, it was in the 2 to 3 range, depending on what you were doing with it. So that's the standards. And then what we know from interviews with Dr. Shi at the Wuhan Institute of Virology is she was doing all of her coronavirus work, all of her SARS-related coronavirus work. There's coronaviruses, which is like this big family. It would be like primates. And then there's SARS-related, MERS-related, and 2 others. And so we want to focus only on the SARS-related at this point in time.
Is that the clades? That's clades.
No,
correct?
that's subgenus.
Subgenus. Okay.
So the genus is beta coronaviruses. So there's beta, gamma, delta, and then the subgenus is Sarbeco, SARS-related, MERS-coro, I don't pronounce that right, which is MERS-related, et cetera. So she spoke that she was using SARS-related viruses at BLSA2, which is like a big red flag. And then the other, bit of data evidence that we would bring into a court if we were going to do it that way is that in 2018, the Washington Post actually published some cables that were either leaked or deliberately given to them. I don't remember the details of that, but anyway, where embassy people, maybe scientists, maybe not. Yeah, I think it was a science related anyway, from Beijing, finally got into the Wuhan Institute of Virology and, met the people there. And they pointed out that there were training deficits in the personnel, which were significant, and that there were practices and facilities that looked substandard, and that they sent, just a letter back to the State Department covering their own, whatever, but nonetheless saying, we think this is a potential problem, and it just, it went into the State Department vacuum, and it's like the end of that Indiana Jones movie where things are rolling through this huge warehouse.
Yeah, the State Department, they have an econ section and a political section. They've got various responsibilities, and usually, it's not 100%, but usually there's a science and technology, either individual or individuals that handle those sort of things, everything from intellectual property rights to these sort of lab things. So there's probably somebody in the econ section over there in China who, was assigned, hey, listen, this is your portfolio and have at it.
Yeah.
Okay. So taking a step back, you mentioned earlier about backbones. Do you mind defining what you mean by backbones for folks? Because a lot of people don't know what that is. And then,
great question.
Yeah, go for it.
That's a great question. And again, there's obfuscation by some of the virology community to, make a straw man argument and then blow it down. So when we talk about making a synthetic virus in the laboratory, right, we are talking about less than 5% of a virus. There's no way, no scientist in the world yet can take a white piece of paper and write the 30,000 letters ATGC in some order that will create a virus that will grow. We're not that smart. So what is done is a backbone is taken from nature, and that's and it shouldn't be called a backbone. It's a virus that works. Is usually the criteria. Although they can be viruses from feces that don't actually work, but have the sequence of something that would work. But anyway, you start with 30,000 letters, and then you play with 300, 400, 1,000 or less of that in your manipulations in the laboratory. So that's why when I say that one of the challenges for a natural spillover is all of the viruses that recognize ACE2, which is what SARS-CoV-2 does, which is what SARS-CoV-1 does. All of those viruses start to appear in nature about 1,000 kilometers south of Wuhan, and then they go down into Laos. So Hubei, the province where the Wuhan Institute of Virology is, you can imagine if Wuhan Institute of Virology has been sampling viruses from nature for 2 or 3 decades, they've probably just, driven their car or taken the bus or the subway to their local wilderness areas. So Hubei province is heavily sampled by the Wuhan Institute of Virology for SARS-related viruses. And they have the unusual property of having 2 deletions in their spike protein that absolutely eliminate their ability to bind to ACE2. So when Dr. Shi heard that this was a SARS virus showing up in Wuhan, and she was on a train, I mean, she reported in Scientific American that one of her first thoughts was, gosh, why is this happening here? Because these viruses are from southern China. Her second thought, which she recorded, which she probably regrets, is, gosh, could this have come from my laboratory?
I remember reading that and thinking it's a natural thought for most people. And
it would
to
be a
go
natural thought only, though, if you've been working on things that are that could
Right.
do this, right? If you're
Guilty knowledge.
completely innocent, yes, exactly. If you're completely innocent, you wouldn't necessarily think that.
So when you talk about going out and picking these up, I've heard it called longitudinal studies, and they had large scale longitudinal studies that, as I understand it, funding came from the PREDICT program through EcoHealth Alliance to help fund WIV's longitudinal studies to collect up these viruses. Can you talk a little bit about why they do that? Why it was viewed important to go out and collect these?
Yeah. So again, let's talk at the highest level. So we'll talk at 80,000 feet, and then we'll talk about 2 airplanes at 70,000 feet. But the 80,000 feet version is a hypothesis that by understanding what's out in nature or manipulating what we find in nature in ways that evolution could lead to, we can get ahead of a pandemic. And I think that's a completely false hypothesis. I can give you a lot of evidence why it hasn't worked. It obviously didn't work in SARS-CoV-2. But nonetheless, that's the operating hypothesis of a lot of folks, including, the Gates Foundation, EcoHealth Alliance, NIH, PREDICT, all of these people who think that they can get ahead of the next pandemic. And there's so many reasons why that's just a false premise.
Did it ever work?
No, it's never worked, and it's not.
That
The
sounds
science
like
so
government
much
work.
shows that it would never work. Let's go back to that experiment where Ralph Barrick and Dr. Shi purposely serial passage a MERS virus to cause it to change species. It changed 4 letters out of 30,000, and even knowing what those 4 letters were, they could only predict why 2 of them occurred, and the other 2 are a mystery to them. So the concept that you could do something in the laboratory that would actually predict the next pandemic is ludicrous, absolutely ludicrous. The other side is, well,
go ahead.
we now know that if you know a virus sequence on a Friday, you can turn your machines on over the weekend, come in Monday, and you can have the raw materials of an RNA vaccine, and you're off to the races.
Or at least the precursors, so you can actually do the research to maybe build that vaccine.
Yes, yes, yes. I should have said that better. You're exactly right.
Well, no, but for me, well, I have feelings about how long we should probably spend researching these things. But let's see. I'm going to skip the next one because you've already covered the piece related to that. But so they had these backbones. They collected them up. Can you talk a little bit about how they make this scientific data available for other scientists to be able to do work?
You mean how they make it available sometimes and they don't make it
Sometimes.
available other times?
Yeah.
Yeah.
Sorry, you know my leading question here, but okay.
Yeah. So that was the 80,000 foot hypothesis. Hypothesis is that's why they're doing this work. There's 2 approaches they took that are 2 planes at 70,000 feet, and we've talked about the gain of function research where you deliberately change a virus in the laboratory. But what they were doing by going to bat caves and collecting samples, and at one point in time they said they had 22,000 viruses they had collected from Asia, mostly China, but Asia, 22,000 in a database. I frankly call that a gain of opportunity, because if you're going into a cave that's visited once or twice a year by a person, collecting viruses that are in a mixture, so they collect feces, from a bat. It's a pretty gruesome process, but anyway. So your sample may have 30 viruses or 50 viruses. So each virus is competing against itself. You take that you take that swab, you bring it back to a city of 11,000, you in your laboratory that's on the subway system, and you purify each one of those viruses, you've created what I call a gain of opportunity. So now this virus, if it, instead of having a chance to infect one person once a year, or every 6 months or to a year in a cave, they get the chance to break out of a laboratory in a city with 11 million. That's a massive gain of opportunity in and of itself, whether you manipulate the virus or not. So both gain of opportunity, gain of function, to me, are problematic if you don't believe the hypothesis that we can predict the next pandemic.
That answers the question. They had this information, they pull it out, they put it in a database. Are you able to talk a little bit about our access to that database and the information? Because don't quote me here, is it GitHub or something like that? Was some of it available on GitHub, or do I have that?
No, you do. There's probably 5 or 6 chapters in the story of data being available or not being available. I'd like to start with the first one, which again to me is so primary. So at 1 a.m. on September 19th, 2019, the world had access to the Wuhan Institute of Virology virus database with 22,000 viruses. 2 a.m., it went away, and access has never been returned. We know that internal what do you call them, IP addresses inside of China were sampling that September, October, November, December, January, and then even that stopped. But I'm not an IT tech guy, but the people I work with are, and they're part of the Drastic group and all of that. So they were able to show that the server was accessible until 2 a.m. on September 19th, 2019, when it stopped being accessible and never was accessible again. I don't have proof that Predict or EcoHealth or NIH monies went into that collection, into that database, so that we had a monetary interest as a nation or whatever in that. Seems highly likely, though. So that access is really problematic, because when you look at the genome, and again, getting back to this deep analysis I've done, is probably looks like August or September is actually the first case. That's probably the day when the animal infected the first human, and the rest is history, as they might say. But so that's problematic. Now, then there's what you call the GitHub issue, or the gene bank issue in the United States, where folks have seen virus sequences from like human patients in the January-February timeframe put inside of Gene Bank, which is the NIH repository for genetic information. You're required to put things there if you're going to publish papers. You're required to put things there if you're getting NIH money. So not putting it there is a violation of NIH money issues. But anyway, so doctors in China with patients collecting specimens with SARS-CoV-2 sequences, January, February, put it up there and then took it down. And then an investigator from the Fred Hutch in Seattle happened to think, well, gosh, maybe it's somewhere on the internet, kind of like the internet never forgets. And it turns out that the NIH was, like most people with large databases, renting space in Amazon's web servers, and they had deleted it from the gene bank in whatever Bethesda or wherever those are at NIH, but they hadn't deleted it, at the Amazon web servers. And so this investigator at Seattle, at the Fred Hutch, was able to download some of these sequences that had been, quote, deleted, unquote, from January, February. And they had very interesting information that pointed to issues around the origin that were important. And so, I think that the train of thinking is, these were put up there innocently in the scope of public health scientific interest. Then parties start to say, hey, these sequences have things that could point to a laboratory. Let's take them down. They weren't properly taken down, so they were accessibility. And now I and everyone in the world can see that, yes, these things tell us things about the virus that probably point to earlier origins in the fall, multiple of this lineage A, lineage B being an important distinction, that the market never had lineage A, and therefore the market cannot be the genetic source of the virus, et cetera.
Great. One comment and then follow-up questions. The comment about Drastic. I have been following Drastic from the very beginning. And I'll tell you, just honestly, if I were to go out and look for investigators in a professional, in my former professional capacity, I would
Yeah.
have chosen that group. 100%. Those people, can you imagine what they could have done if they had the full weight of and resources of the federal government to go do what they were doing? They were on the ball. They came up with the Mojang minor stuff. First people to come up with that stuff which I didn't get into, but we can later. But so yeah, I love what Drastic has
I
done.
published about 15 papers with them.
Mhm.
I was asked to join. I chose not to. It just they're extremely bright. They're extremely clever. They have, of course, they have Mandarin. There many of them are Mandarin
Right.
primary speakers.
I was checking their translations. I too speak Mandarin, and I was really, they're very impressive. Very impressive. So sorry, I want to jump into a slightly related topic. So you've got the Wuhan Institute of Virology there. There's other facilities in Wuhan, though. There's the Wuhan Institute of Biological Products, which, and then there's, as I understand it, there's even some pharmaceutical industry type companies that are in the larger area, maybe. Can you talk a little bit about, could some of this work have been done in, say like the Wuhan Institute of Biological Products that have, as I understand it, don't they have animal testing facilities there? And is there a possibility this sort of stuff could have occurred in the related facilities? And what does that mean?
Yeah, again, you're exactly right, and I'm going to be very careful here. So while I can conclude with 98% probability it came from a laboratory, and with high probability it came from the Wuhan Institute of Virology because of the genetic manipulation, which is unique to that institution, you are absolutely right. So, for example, Wuhan University, which is, I think, 2 kilometers from the old WIV facility, slightly north and on a lake there, was a subcontractor of the WIV, which is a subcontractor of EcoHealth, which is a subcontractor of, or which is a direct contractor of NIH. And they had monkey facilities, which they had agreed in some grant proposals to test some of the viruses that were being created at WIV and that sort of thing. So the fact that now there's one of the earliest patients that I found at the PLA hospital, which is also like 3 clicks from the Wuhan Institute of Virology, has her 23-year-old young woman, her what do you call it? Coordinates, her latitude, longitude coordinates, where she, quote, lived, unquote, were at the Wuhan University. And some of the off the record information I've received, which indicates that that may be the source of the infection. You want to check that box. You've talked about the Wuhan Products Company, which is a vaccine company doing vaccine research, vaccine development, and they're right on the river. They're a little bit west of Wuhan Institute of Virology, 2 subway stops on line 2 further west. And they, in the WhatsApp discussion groups that were tracking people talking about virus, infections and that sort of thing, they are the focal point within half a kilometer of focal point of some hotspots in early January as conversations were happening. They had dormitories there where both their own employees and employees that were down at the BLSA-4 WIV facility lived. And there was a daily bus that went back and forth between those dormitories and the BLSA-4 WIV facility about 15 kilometers farther south. So that's another potential hotspot. And I think the last one is probably the Wuhan CDC, which is 100 meters from the market, and which moved in the November-December timeframe from another facility. A known risk factor in samples, contaminating people is when you move a laboratory, right? So you got a refrigerator full of deadly pathogens, that you never open or you open once a day. Okay, now we move everything from the refrigerator into into cold packs, and we put them on a bus, or put them on a truck, and we drive them across town, et cetera. You can see the picture there. The risk escalates massively when you move a laboratory, and that happened in November, December 2019. So there are many other potential either collaborators, co-conspirators, suspects, and I want to be careful not to fully indict
Right.
WIV, but the genetics inside SARS-CoV-2, the skills, both published skills, know-how, patents, are primarily there. I mean, another interesting side note: November 20th, 2019, WIV publishes a patent on a little tourniquet that you can put on if you get bit by an animal to prevent you from getting a viral infection. They published that in November 2019. And
I remember hearing that. Yeah.
Yeah. So I mean, I was the one that found that and translated it from Mandarin to English. So, necessity is the mother of invention, and so that's kind of suspicious for someone getting bit in the fall.
Yeah, I remember reading. It's funny you bring that up. I hadn't thought of that in a while, but I do remember reading that. That was your publication.
Yeah.
Oh, wow. Okay. Fantastic. The reason I bring up some of the pharmaceutical industry stuff is, recently, and I'm jumping ahead, but, Cadlick recently stated something to the effect of that there was vaccine research going on, and this is a product of vaccine research. And on a sort of I don't know what level, on a level, it makes sort of sense because a lot of what our NIH and NIAID were interested in the past was vaccine related research. So it makes sense to me on one level that perhaps there could have been collaboration. But let me keep jumping forward because I want to utilize our time to the best of our ability here. Can you talk a little bit about, in layman's terms for everybody, what it means when a newly emerging virus shows low genetic diversity? What are the epidemiological implications of a low genetic diversity popping up with a new virus that just pops onto the scene?
So I'd like to go back to remind you of what a zoonosis is, because when you think in those three buckets, animal, virus, person, and then you look at evidence in each of those buckets deliberately, it really becomes informative. So just to check the box on the animal. So there's 80,000 animals tested in China. No animal ever had the virus that they ever found, and no antibodies antibodies from SARS-CoV-2 were found in any animal anywhere. So the market, the farms that delivered the markets in Wuhan, other places in Hubei province, every province of China, 80,000 animals, 400 species, 0 viruses in the wild. So the animal is missing. SARS-1 and MERS, in the animals in the market in SARS-1, 100% had the virus. MERS came from camels to humans. 100% of the camels in the markets in the Middle East had MERS. So we got 100%, 100%, and 0 out of 80,000. Check that box. In SARS-1, because the virus had spent, it looks like about 3 to 4 years jumping from bats to civet cats in the markets, the virus makes a mistake every 2 weeks, makes 26 mistakes a year. So you can with computer equations, you can say almost to the month when the virus first jumped from a bat to a civet cat, and then started to spread in the civet cats environments. So now it's saturated the civet cats. It's spent 3 to 4 years in the civet cats doing that, and now it starts jumping to humans because it gets that additional mutation that's good for jumping to humans. All of those get that. So when you look at the first 100 infections in humans from the market and from SARS-1, they have a lot of background diversity because the virus is making 26 mutations a year in the process. So what's called posterior diversity, which is diversity, so that the early human cases in SARS-1, the early human cases in MERS had a lot of background genetic diversity. So they look like if you built a family tree, it doesn't go back to the month the thing happened. It goes back years earlier. was violated with SARS-CoV-2. SARS-CoV-2 came from one virus, and so every infection, all 1 billion infections in the world, are all from sort of one atom, one individual virus that started. And that's never been seen in a natural spillover. It's never, and the fact that one, and it's still, when I say it, I still astonish myself. One animal infected one human, and we have 1 billion to 2 billion infections as a consequence of that is just mind-boggling. So
So
that checks the box on the virus. If I could just finish, so I have all
for
three of
sure,
them
for
for
sure.
you. So all of the scientists that were saying this was a natural spillover said, well, one of the things that you'll find in a natural spillover is what's called seroconversion, which is the finding that in the markets in SARS-1, in the markets in camels with MERS, and in the MERS outbreak, the virus is going to jump to humans, but it doesn't have all the tools it needs to really infect a person, so it could be asymptomatic, or infect a human so it can jump to another human to human transmission. But that human will have antibodies in their blood from having had that infection. So in southern China, once we had antibodies for SARS-1, 2 to 3% of the general population had antibodies against SARS-1. You go into a blood bank, right? People give blood. 2 to 3% will have seroconversion. You take the people from the market, oh my gosh, they're exposed in the market. 20 to 30% have it. Same thing in the Middle East. 1 to 2% have background seroconversion, and people in the market or people exposed have a higher level. So so everybody, all the virologists, zoonosis as I call them, predicted, say, hey, yeah, we'll go back to the blood banks in Wuhan and we're going to find 2%, which is our expected standard. So they go back, they test 36,000 in the blood bank. Jim, I'm going to ask you to tell me how many of that 36 they found were seroconverted. Drum roll, zero.
Uh,
So
uh,
2% of 36,000 is what? It's 720.
mhm.
So you predict 720 out of your samples are going to be seroconverted, and you find zero. You get your statistician to run the probabilities of that, and you're in the 1 in billions. So no animal in nature had it. The virus had no posterior diversity. It came from a single case. And the zoonotic actually admit that. They say this looks like a single transmission. And then there's no seroconversion, which means it wasn't experimenting in the environment to get the tools. It was experimenting in the laboratory. So those are the 3 pillars of a zoonosis, and all pillars point to a laboratory.
So, and I think I know the answers to this. It's been a while, but antigenic shift and drift is what's needed for something to happen, to come out and really like hit a human population and get that kind of infectivity that they're not prepared for. And if you don't mind, for everybody, talk a little bit about antigenic shift and drift.
Sure. So ACE2 is a surface protein in the lungs, in the brain, in the kidneys. It's sort of all over the body, and its natural function is to cut or cleave a hormone involved with blood pressure and water maintenance and that sort of things. So it happens to be the target that SARS-1 used to bind to, and then to get into the lung cells to reproduce to cause an infection. So ACE2 is different in evolution, right? So civet cats, humans, monkeys. I mean, again, the closer you are, the mice. All of these animals have ACE2s, but they're all slightly different. So when a reservoir virus is in bats, reservoir host means it's a host that the virus can grow, and it doesn't cause it's a mutual destruction or something. They've come to an agreement, to an accord, and it doesn't affect the bats very much, and it doesn't affect the virus. It doesn't kill the virus. The bats have an ACE2 that's different from civet cats, so it starts jumping to civet cats because it has an opportunity, but it doesn't infect very well. And then civet cats, it jumps to humans, it doesn't infect very well because the ACE2 is different. So this concept of antigen drift is that it needs to migrate from recognizing the ACE2 of the civets to recognizing the ACE2 in the humans to being good at infecting humans. And then it needs to get around all of the mechanisms that humans have evolved for stopping viruses. That takes time, that takes mutations. So antigenic drift is the mechanism by which Darwin's Origins of Species, his observations of changes in evolution, is done.
Gotcha. So then one other related definition I'm hoping you can help people understand is founder effect. And I think you've sort of described it, when it starts with, sort of a very small subset, but I'll let the expert talk about founder effect.
Yeah, well, I mean, and there's lots of ways founder effect could be used, so I'm not quite sure how you want to use it here, but in human populations, the founder effect is the concept that you use these computers to predict back in time. You can predict within about a 50-year, 100-year period of time when the first mutation that happened in a human that was extremely useful. So two examples, the Sherpas that assist people to get to Mount Everest often don't need oxygen. They're just remarkable folks.
Right.
And the reason is they have a founder gene back, I think, 3,000 or 4,000 BC, which changed the way their hemoglobin interacts with oxygen in a favorable way to allow them to live comfortably at high elevations. And because they decided to adapt to that particular spot, that advantage got passed along. The other is the BRCA gene, which was in an Ashkenazi Jewish background in about 4,500 BC, gets passed along, and that one is associated with a high risk of breast cancer, a high risk of testicular cancer, and I never understood really why a founder gene would be so prevalent that causes this effect. And the answer is that, of course, breast cancer and testicular cancer happen mostly after reproductive age. And the BRCA gene migrates closely, co-migrates with genes that are associated with intelligence. So if humans are selecting their mates, one of the criteria might be intelligence, then this BRCA gene goes along for a ride. And if most people are having their children by 30 or 35, the negative effects of the BRCA gene don't impact its maintenance. So founder effects are something that happened in the past. They give either an advantage or a disadvantage, and they have other effects going along with them. So I mean, in the context of SARS-CoV-2, the first virus that came out had, because of its serial passage, I believe, aspects, it was highly adapted to the human ACE2, to the point where human-to-human transmission was working effectively right from the get-go. And of course, one of the challenges to doctors, medical doctors in the field, in the hospitals, infectious disease docs, is natural spillovers have one advantage for public health, and that is that they're not very good when they first infect humans. So they may kill the first few patients and that sort of thing, but like Ebola and like MERS, they haven't perfected human-to-human transmission. So all of the folks are saying sort of two things at the same time: is we got this brand new virus and we don't know what it's going to do, and it's killing about 10% of the people, but our background and our understanding of science would be that it's not going to be a human-to-human transmission issue. So we're going to have some time to get our act together and go forward. One of my biggest issues, I guess, with what China did in this context, and then eventually what the US was doing in this context, was we started to know very early that human-to-human transmission was happening. There's published papers from the end of December. There's evidence from the doctors talking in China at the end of December, but talking was suppressed, that human-to-human transmission was happening in an extraordinary way right out of the box. And whether you worry about origin, you want to understand that, because that has huge public policy implications. And we lost about 6 weeks, and we lost transmission around the world. And again, because those events early on have an amplification effect, we could be talking about 500 million infections that might have not happened had we had a 6-week jump on knowing human-to-human transmission was occurring.
Right. When you can't put the toothpaste back in the tube. So, we talked a lot about animal reservoirs, and I want to jump past this here in just a minute. But I've heard people talk about minks, civets, raccoon dogs, pangolins. I think I even read something about some sort of rat, like bamboo rats or things like that. Um, from the sounds of it, though, the physical habitat of where these animals would need to be and co-located just simply wasn't. It wasn't in Wuhan, and it wasn't in the Huanan market, the wet market. Is that the data we have? Is that correct? That
Well,
geographic
that's correct, Jim, and
overlap?
yeah, let me talk about this because there's two important pieces of evidence that you need to understand to again kind of cancel out the idea this came from nature. So one is three, I should say, not two. One is the finding that when you look from nature at all of these species that are potential hosts, you find zero infection and zero seroconversion. So you don't find any animals that are infected. Number two is when you try to infect these animals in the laboratory, the infections don't stick very well. But number three is a finding about the virus that when it first came out in humans for about 6 weeks, it had a particular mutation in its spike protein, which didn't stabilize it with the furin cleavage site. And this mutation is called D614G. So the D version of the virus, the spike protein actually falls off. The virus becomes blind, and it's only about 10% infective. So this mutation happened in humans in January for the first time. It happens every time you do it in animals in nature. All the billion infections, for the most part, are the D614G mutation. What's the implication of that? So since it came from nature with the D version, and since we know the D version cannot be passed from animals more than 3 animals deep without changing to the G, there is no theoretical way where the D version could have come from nature if it had been in nature 6 weeks, 8 weeks, 10 weeks.
Well, I want to thank everybody for listening. It's a long interview. It's an hour long, and it's the first in a 2-part series. The intent was to give you some information that goes beyond just the what's going on behind the scenes with the federal government, but sort of broader issues that are of interest to not just federal government employees who are interested in the science, but also the wider public that wants to know a little bit more about the science behind SARS-CoV-2. So here's what you can do to help. Please go to fedsforfreedom.org, become a member, sign up for one of the tiered memberships, or even just donate money. It's a tax-deductible donation, and it will help federal government employees, the only federal government employees, who stood up to vaccine mandates. All of us in our organization said no. Well, most of us. Some of us didn't want to, they had to. They were forced into a decision they didn't want to make, and it was immoral. So if you really want to help, we've got lots of lawsuits that are still needing funding support. And so if you go, sign up to become a member, sign up to donate, sign up for our newsletters at a minimum, you'll get more information like this. So I guess the last thing is we'll be posting this on our podcast in short order. And we'll have the second part of this interview with Dr. Stephen Quay out as soon as possible. There's a lot of ground to cover when you're talking about the origins of SARS-CoV-2, and I personally find it fascinating. I hope you do too. Thanks.